All right, everyone. We are moving on to our first lab exercise of the day. Before I go on, I want to mention, in case people weren't aware, and oh yeah, let me know if this is correct. Lunch is being provided, right?
That's correct.
Okay. And where is that going to be? Right here. Okay, it's going to be in here. Okay, great. So that'll take place at 12. Also, at 3 p.m., I like to do a group photo. If you want to be a 3:30, you want to be a part of history, well, let's go up here and take a brief photo. I just like to document the different workshops that I do. So you don't have to be in the picture, but Instagram, I don't know. I'm not on Instagram, but okay. Yes. At this time, what I want you to do is open up MATLAB, whichever one you have. If you don't have it, weren't able to install it, please share with somebody who does have it. I'm not expecting that everyone has everything installed perfectly. This just happens just by the numbers, but you should be able to still follow along. All right, so while that's getting loaded in the background, let's see. On the schedule website, we have from 11 a.m. to 12 p.m. how not to do it. It's practical. And there's a link to a sample data set. Okay, I'm just going to make that clear. So this is the schedule web page. Okay, this is under day 1. Scroll down. 11 a.m., 12 p.m., how not to do it. It says the sample data set can be found here. Right? So click that link. Let me know if anybody needs more time falling behind. Okay, this is from the SPM website. And if you scroll down under batch, they have this link that says auditory SPM 12 batch. Click on that to download it. Good so far? Okay. Give me 1 second. Don't copy what I'm doing here. I'm just going to make sure that everything is okay. Everything's fine. Okay. So I just didn't want to overwrite something I already had. So if that is downloaded from the MATLAB interface, and so I'm assuming that everything is downloaded to your downloads directory. If it's not, go to where that script was downloaded. But in my case, I'm going to type cd a tilde, which is in the upper left part of your keyboard, downloads. Okay. Press enter, and you should see this auditory SPM 12 batch file. Whoops. So just type the name right here, auditory SPM 12 _ batch, and when you're ready, press enter. So I'm assuming that everybody had installed SPM correctly. I didn't hear anything, so that's what I'm assuming. If there's a problem, again, don't freak out. Just find somebody who does have SPM installed and work next to them. What this is doing, this is pretty good on the SPM side. They have a script that's going to automatically download a very small data set, and it's going to do a start-to-finish analysis on that. So things like the realignment, pre-processing. Pre-processing is something I'll get more into when we talk about the Con toolbox, because that also requires these same pre-processing steps that we're doing right here. So you'll get a more in-depth overview of what's actually going on. But the point is, I want us to go through this data set, and it's going to do a very simple contrast, auditory contrast. So this was a data set where I believe people just listened to some auditory stimuli, then there was a period of rest. Again, very simple. I'm using very basic examples, but my intention is you can use them as a surrogate for your particular experimental design. So this will take, I don't know, 6-7 minutes. But in the meantime, just let that run in the background. Anybody having any problems? This is scary how silent everybody is.
You've already had SPM and it's not your download. Is there a specific area you should go to do this? Like SPM folder?
Oh, if you want to run it in another directory, that's totally fine. Yeah, you can just move it and then run it there. Yeah, yeah. Sure, yeah. It really doesn't matter. I mean, if you don't want it to get too crowded in a certain directory, you can make your own, put it in there. I'm just assuming that most people just by default go to downloads, and that's where I'll be running this as well. But going back to this. So this is we're going to try a few things out here. I'm just going back to my slides. Remember when we were talking about these biased ROIs? Let me find the figure I want. Well, let's just use this one, for example. So instead of these, ROIs highlighted in red, another common way to create an ROI is from the data itself. And people might reflexively say, oh, it's biased, don't do it, it's bad. Well, yeah, if you are extracting data from that same data set, right? Somebody came up with a question, what if it's an orthogonal contrast, statistically defined, as in there's no variance shared between them, or if it's data from another scan, for example, same subject, different scan. That's also valid because that data is not going into the actual analysis. But if you are using it with the data you're eventually going to do inferential statistics on, it is biased. And when I first was learning about this, people said, don't do it, it's bad, just don't do it. Well, we're going to do it because it's fun. And how else are you going to know if you're doing it wrong unless you actually do it wrong in a very safe environment? We're not going to publish this, we're not going to make any wild claims, but I want people to know how to do it. And it's fun because you'll see your effect sizes just go through the roof, and it's awesome. It makes me feel really good to see that. Let's see here. This will still take a few more minutes. Yeah. So in the meantime, since I have a little bit of time here, we're not going to get through. Oh, it's almost done. But let me just present a little bit on this. This was one more insidious statistical problem that catches a lot of people off guard, and this is something called parametric modulation. Now, some people may never even design a data set that has to do with this, but in a nutshell, parametric modulation means that for a given trial, there's certain parameters that can be modulated or changed about it, right? Say I'm flashing a checkerboard at somebody, and the checkerboard is the condition, but I can also modulate the brightness, let's say. So I have the checkerboard condition, but I have 5 different levels of brightness. And so for that given trial, does the amplitude of the activity I'm eliciting vary, let's say linearly, with the amplitude of the brightness, right? Other examples, in gambling studies, this is very common, right? So you have a certain gamble, and there's a certain, say, expected value associated with that gamble. I did something that's pretty recently. Is a pretty interesting kind of multi-site study. We're all trying to replicate each other's analyses, but it was a simple data set. It was a gambling data set, and for each gambling trial, there was an expected value. You do parametric modulation on that, and that seems to load on these reward-sensitive regions. Expected value goes up. Nucleus accumbens activity tends to go up. If it goes down, it tends to go down in that area. But just a few words on this. If you're using SPM, which a lot of people do use SPM, if you do parametric modulation, be aware that the order of the parametric modulators you put in there does matter due to something called orthogonalization. So here's an example. This was from a study I did back in graduate school before I became aware of this. If these are glass brains, just so you know, don't worry about it too much, but these in darker, these are the actual significant clusters. But here was when I put, let me put my turn signal on. You can enter multiple parametric modulators for the same condition. So if it had something like a gamble with expected value, and say loss magnitude. I mean, ideally they should be, pretty orthogonal, but sometimes they're not. If I put that, say I put, let's work with that example. I put an expected value first. It's my first parametric modulator. I get this really nice constellation of clusters. If I put it in second, if I put, say, loss magnitude first, expected value second, for some reason it totally dies on the vine. Why is that? It has to do with something called orthogonalization. So this is a big problem because some people put in one first and they'll say, oh, that one's significant, the other ones aren't, but there's something else going on here. This is from Jeanette Mumford's 2015 paper. So think of orthogonalization as assigning pi to different regressors. So if I have, say there's a trial with, I don't know, the intensity of the checkerboard, and there's also the subject's reaction time on this trial. I can modulate their activity on that trial by both of those. Let's say that, and the unmodulated is referring to the activity for, say, just the checkerboard, but the parametric modulator is anything kind of above that. So you're always going to have this unmodulated one referring to just the regressor everything else is attached to. If I orthogonalize, say, reaction time with respect to the unmodulated regressor, you see that all three of these things share some variance. But if I orthogonalize it, and I say orthogonalize the second parametric modulator with respect to the first one, basically any overlapping variance is going to be assigned to the one that gets put in first and not the one that gets put in second. And I can flip this around and say, well, if I do this and I orthogonalize reaction time with respect to intensity, intensity gets a greater share of the pie. So it's really an artifact of how you're orthogonalizing these two things. The correct thing to do would be to leave this shared variance out of the equation, which is what happens here. Again, we don't have time to get into all the details about how to do it, but I can give you, I'll give you a resource showing you how to correctly do this in SPM because the defaults will not do this for you. Does anybody do parametric modulation? If nobody does, okay, a couple people. So this is something to what you use AFNI. AFNI is fine, which is great, yeah. AFNI does not have this problem. I've tried it. It doesn't matter what order you do. It does the correct orthogonalization. So just be aware that you do parametric modulation with SPM. We can talk more. Okay, at this point, is anybody's analysis still running? Yours is still running?
I had an issue with the file.
Okay. 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 But everybody else sees this window right here with the surface plane and everything. This is a rendering of the results onto a surface. Just in case you were wondering, it makes for a prettier picture. You can kind of see where it's tracing the gyri and sulci. There's a more correct way to do this. We don't have time to get into using FreeSurf and all that, but you can find it on the book. It's if you're so inclined. All right. So we have this beautiful auditory signal in both the left and the right auditory cortex. Again, if you're new to SPM, just bear with me. It might go a little fast. Other people, this may seem really easy. But what I'm going to do is click and drag this. Can I click on it? Come on. I used to be able to do that. Never mind. Click on whole brain right here. There's another way to do this. I apologize, it's a little tiny here, but there's this other panel. Let me clean this up just to make it easier to see what's going on. This panel right here, whole brain, and you'll see a list of all the significant clusters. SPM does its own cluster correction mechanism under the hood. I'm a little wary of it. I prefer to do my analyses outside of it in something called 3D Clussim. I talked to a couple people about that. Again, I can provide the code for it. In any case, let's assume that these are, in fact, corrected for multiple comparisons. If I click on these coordinates over here, right, they'll snap to wherever that peak voxel activation is within the cluster. So click on this second bolded line right here. This is the left primary auditory cortex. And these have been thresholded with a voxel-wise multiple correction threshold, so that Bonferroni correction of 0.05. So it's a very strong effect, and you can do that. You can get away with that with these really robust, strong effects. OK, so if you have that highlighted, you can save out that cluster as a mask. So there's a drop-down menu down here, overlay, and if you go to current cluster, oh, sorry. If you click on save, click on the save drop-down menu and click on current cluster. It's going to prompt you to give an output for the file name. And I'm going to call this left odd _ 05. Doesn't matter what you call it. I'm just indicating this is the left auditory cortex with a correction level of 0.05. And if you press enter, you will see in the MATLAB terminal that this has been saved to a file, left odd 05. Again, you don't need to follow along with this. I have a bunch of stuff installed on here. But just to show you how that mask has been saved out. This is, I love FSL's viewer fossilized. So in white here are all those voxels. They've been binarized, so it's simply indicating one where there's one of those voxels belonging to that cluster. And I can also add a standard here, a template, because it's all been normalized. Let's do something like that. Notice that left is on the right-hand side in this FSLized viewer. OK, so that is my mask. This isn't just another way to look at it. And if we do an ROI analysis, we're averaging across all the voxels within that ROI and then doing a test on it. It's a single subject, so we can't do any kind of inference, but we can see how the average value changes. Now, if you go back to the schedule website, you will also see another highlighted thing called, we can do some data peeking using this script. Just click on it. This is on my GitHub. You have a couple options here. You can copy and paste it into a M file. These are MATLAB files that run a block of code. So this is all MATLAB code in here. What I'm going to do is click on raw and then right-click anywhere in this panel right here and click Save As. If you're a GitHub guru, you can do this another way, but this, for most purposes, this works just fine. Remove that text extension and save this file. By default, it's going to call it extract ROI data.m into the GLM directory. So this is saved all of your output from running like a basic auditory contrast, right? Simply save it within there. Now, I'm using this in this case for a bias ROI analysis, but you may find this useful on your own if you want to use this for an actual ROI analysis. It takes two inputs. One is the ROI you want to use, and the other one is the contrast that you want to use. That looks delicious. OK. Let's hurry up. Come on. Don't want it to get cold. Let's see here. OK. Anybody have trouble with that? Everybody has that file downloaded? OK. You got to understand, I keep asking this because in some workshops, this is like total chaos, and you got to run around it. But you guys are awesome. It was at people were having a little bit of trouble. It's okay, different places are different. Okay. So we have this extract ROI data command, right? So actually, just let me show you what's in here. We have, this is our ROI, our mask, and this con 001 is the contrast for, I think it's auditory minus control, right? So to use this script, extract ROI data in parentheses, and then in a single quotation to indicate it's going to be a file, type left odd05.nii. You can also, if you want to remove the guesswork out of it, you can copy and paste these things into there as well, whatever you want to do. Again, to repeat, this file will simply average across all of the voxels within that ROI for that particular contrast, for this auditory contrast. This is what I'm talking about when I say, if you want to unpack a main effect or an interaction, this is how I would extract the individual contrast parameters for that. So you hit enter, you get a value 1.96. I mean, what is that exactly? So SPM is going to estimate beta weights for the condition of auditory and one for control, I believe, and just simply take the difference between the two. In this case, anything with a primary sensory area, like the auditory cortex, we're going to have a really big, robust response. So something on the order of 1 or 2 for a contrast estimate is reasonable. For most experiments, you wouldn't find something this high. It's pretty rare. But this is kind of the ballpark that we're looking in. One more related thing, just to kind of try to tie all this together. When I talked about orthogonalization, right? If that, in a sense, removes the shared variance or the correlation between regressors. If the regressors are really highly correlated, this is kind of a separate topic but related, you're going to have a lot of shared variance between them. And if that happens, when you try to estimate the beta weights for them, they're going to be very, very high on the magnitude of like 7, 8, 10, 15 sometimes. That's an indication that your regressors are highly correlated. Just another kind of sanity check. But in this case, it looks pretty good. Okay, so ROI analysis for single subject, pretty easy. Now, I want to go back to this control panel right here. And if you click on this menu, contrast, we can change the significance level, right? So it's just, originally it's at 0.05 FWE, is what we just had here. Let's click on change and set this to something else. So again, FWE, but let's pick a different threshold. Let's just poll the audience, or whoever's the loudest. What threshold do you want? It could be anything. 000, that's, except for that might be too strict, but what the hell? Nothing ventured, nothing gained. 001, is that correct? And no cluster extent threshold. This is a voxel-wise threshold. Oh, wow, that's a very strong effect. Okay, very good. If you click on, say, current cluster, okay, it's going to highlight this left auditory cortex again. If you remember, how does this look different from the one we had before? A little bit smaller, right? Obviously, because we've removed more of those less significant voxels. Now, we're going to save this one out as, again, save current cluster. Let's call this left odd 001, or whatever you want to call it. Press enter to save it out. And here's a MATLAB shortcut. If you go back to the command line, back to the terminal, if you press the up arrow, it'll display the last command that you typed. So this is useful because I can simply go over here and just change that to 01. Don't press enter yet. Don't press enter. We're going to think a little bit. Before we got a contrast estimate of 1.9 something, right? We've now made our threshold for what we include a lot stricter. Is that value now going to be higher or lower? And why? Who wants to venture a guess? Higher, lower? Oh, sorry. You think it's going to be higher? Okay. Anybody think it's going to be lower? There are fewer voxels, right? Okay. It is higher. Correct. Yes. So let's think about that. Why exactly was that? Yeah.
They're smaller, they're less voxels, and if they're higher,
Yes.
they will be higher.
Exactly. Yeah. So yeah, the answer was there are fewer voxels. Obviously, the threshold is stricter, but all the only voxels that remain are going to be even more significant. So we keep bumping it up, and you can keep doing this. You can play around with this all day, and you'll find invariably as your threshold goes up, the effect size you're going to get is also going to go up if you do a bias ROI analysis. It just keeps spiraling upwards, and this is what a lot of people do happen to do. Okay. If we stop this a little bit early and eat, and we start the next session a little bit early, people going to be happy with that? Okay. Let's do it. Let's reconvene at 12:40 to then begin the next section.